So hello everyone, this is River and today we are going to do SolidsLab. But before we get started, I would like to introduce some of the background. Suppose we have a wastewater sample and we want to measure the solids content in this wastewater sample. So first, we can transfer some of the samples into a container such as evaporation dish. For this container, we have all the solids in it. And of course, it contains water. So we bake this sample at about 100 Celsius degree. And during this process, water gets out. And we have some residues in our sample. And these residues are called total solids, noted as TS. And then we can further use the muffle oven to bake the sample at about 550 Celsius degree. After this process, we still have some of the residues in the dish. And these residues are called total fixed solids, TFS. On the other hand, we also have some solids that get out from the residue, and they are volatile. So these solids are called total volatile solids, TVS. So these are the untreated wastewater samples. On the other hand, we can also filter the sample, and after filtration, we have two parts. The first part is the filtrate. First, we have the filtrate in a container like shown here, and the filtrate contains dissolved solids. Second, we also have some residues on the filter, and these residues contain suspended solids. Then first, let's bake the filtrate at 180 Celsius degree. And after this process, we have some residues in the dish, and these residues are called total dissolved solids. And then we bake this sample at 550 Celsius degree in the muffle oven. And after baking, we also have some residues in the dish, and these residues are called fixed dissolved solids. On the other hand, we also have some solids that get out from the dish, and they are volatile, and these parts are called volatile dissolved solids, VDS. Now let's turn back to the residues on the filter. Still, we can also bake the filter in the oven at about 100 Celsius degree, and the water is removed. And after this process, we still have some residues on the filter, and these residues are called total suspended solids. Again, we can move this filter into 550 Celsius degree muffle oven to bake. And also during this process, and we'll still have some of the solids on the filter, and these solids are called fixed suspended solids. And we still have some of the solids get out from the filter, and these parts are called volatile suspended solids, FSS. So all of these above are the diagram of solids in the water. So in summary, we use different treatments to deal with the sample, and then we can get different results. Anyway, by knowing this above, we have these equations, and these equations are based on the mass balance. So let's do the experiment. Now let's do the first experiment, total solids. So before we do experiments, we need to transfer some of the wastewater samples into the beaker. So first, this is wastewater sample. And before we take the samples from this big bottle, we need to shake it to make sure the wastewater sample is mixed and representative. So we need to mix it first and then get some samples. And then we need to make sure it is well mixed in the beaker. So for the first experiment, we need to transfer 15 milliliter of wastewater samples into the evaporation dish. And so all of the dishes or the containers, they are in the desiccator to make sure there is no water vapor gets into the container, which will introduce some of the errors. So first, we need to measure the weight of this evaporation dish. And then write down the mass. And then take 50 milliliter of samples into it. And then we bring this sample to the 103 Celsius degree oven. We need to use the tweezer to put the sample into the oven. And then we wait for 54 minutes. But before we take the sample out, we need to prepare the desiccator to make sure there is no water vapor gets into our samples or our dishes. And so when we get the sample, we need to make sure it is cooled down for about 10 minutes. And now it is after 10 minutes. We are going to measure the weight of this and write down the mass after 103 Celsius degree. And after this, we are going to bake it in a 550 Celsius degree muffle oven. So this is a muffle oven, but when we put the sample into the oven, we need to wear gloves. It's very hot inside, so we need to transfer this into the oven and bake for 50 minutes. Now it is after 50 minutes, so we are going to take the sample out. Still, we need to make sure the sample is in the desiccator because we want to prevent the water vapor gets into our sample or our dish. And then this dish would be very hot, so we need to wait for 15 minutes. So now it is after 15 minutes. We are going to weigh the mass of this dish. And write down the weight of this dish. So it is a mass after 550 Celsius degree. So we have the data for the total solids. And then next, we are going to do dissolved solids. So now we are going to measure the dissolved solids. So at first, we weight the mass of dish. And after weighting the dish, we are going to store it in a desiccator. And the next step is filtration. So this is our filtration state. And when we open the cap, it can create vacuum. So before we use it, we need to rinse it. Now we are going to transfer 50 milliliter of sample to this filtration system. And we need to make sure there is no residues on the top of the cap. And now we have filtered it for 3 minutes, and then we get the filtrate. And then we bake this sample in the 180 oven. And we bake it for 54 minutes. So now it is after 45 minutes, and then we are going to take the sample out. And still, we need to wait for about 10 minutes to for the dish to cool down, and then we measure the weight of the dish. And after that, we are going to put the dish into the micro oven. And now it is after 15 minutes, and then we are going to get it out. And now it is after about 20 minutes, and then we are going to measure the weight of this dish. And write down the mass. And that's it for the dissolved solids. And next we are going to do the suspended solids. So now we are going to do the suspended solids experiments. And so first we need to measure the weight of the aluminum plate with the filter. So we measure them together. And then we store it in the desiccator. And so the next step is filtration. And then after 3 minutes, we are going to take the filter. And now we are going to put the filter with the aluminum plate into 103 Celsius degree oven to remove the water. So now it is after 45 minutes, and then we are going to take the sample out. And then after 10 minutes, we are going to measure the weight of the aluminum plate with the filter. And then we put our filter with aluminum plate into the micro oven. And now it is after 15 minutes, and then we are take it out. And still we need to make sure it is cooled down for about 20 minutes. And after 20 minutes, we are going to measure the weight of the filter with the aluminum plate. So this is for the suspended solids. And now we have all of the solids data. And then based on this data, we can do the calculations. Now we are going to do to measure the turbidity. And this is the turbidity meter, and this is a vial for turbidity meter. So we are going to transfer some of the samples into the vial and then use the turbidity meter to do the measurement. But before we take the samples, we need to use the sample to rinse the vial. And after rinsing, we are going to take about 25 milliliters of samples into this vial. Now we are going to use the turbidity meter to measure the turbidity of this sample. But before we put this into the turbidity meter, we need to make sure that it is mixed, because when the solids gets precipitated, the turbidity will decrease. And now we have the reading, and we can write down this reading. And that's it for turbidity. So finally, we are going to measure settleable solids. So for the settleable solids, we are going to use this cone. And in this cone, it has a 1 liter mark here. So we need to fill the column with the sample up to 1 liter. And before we take samples, we need to shake it. And then we let it settle down. So now it is after 1 hour, and then we can get the reading of the column. And we can see this is 2 milliliter. So that's all for the SolidsLab today. Thank you very much.